Inside nearly every cell in your body lives a population of tiny structures that most people never think about, despite the fact that without them, you would not be able to move, think, digest food, or even keep your heart beating for more than a few seconds. These structures are mitochondria, often described in basic biology classes as the powerhouse of the cell, a description that, while a little simplistic, captures something genuinely important. Mitochondria are responsible for converting the food we eat and the oxygen we breathe into a usable form of chemical energy called ATP, the fundamental energy currency that powers essentially every process in the human body. Over the past 2 decades, research into mitochondrial function has expanded well beyond basic cellular biology into becoming one of the more significant frontiers in aging research specifically, as scientists have increasingly recognized that declining mitochondrial function is not just a symptom of aging, but appears to be one of its actual underlying drivers. This has led to substantial research interest in how diet influences mitochondrial health, and specific foods have emerged from this research as particularly well supported for supporting these critical cellular structures as we age. Today we're walking through 5 of them, along with the science explaining why each one appears to matter so much. To understand why mitochondrial health has become such a significant focus in aging research, it helps to understand a few key things researchers have discovered about how these structures change as we get older. Mitochondrial dysfunction has been identified by researchers as one of what are now formally recognized as the hallmarks of aging, a framework used across the scientific field to describe the core biological processes believed to drive the aging process itself. Research has found that as we age, several things happen to mitochondria simultaneously. Their overall number within cells tends to decline, a process researchers sometimes measure through mitochondrial density. Their efficiency at producing ATP tends to decrease, meaning aging cells often generate less usable energy from the same amount of fuel. And perhaps most significantly, aging mitochondria tend to produce increasing amounts of reactive oxygen species, damaging molecules that are a natural byproduct of energy production, but that, when produced in excess, contribute to oxidative stress and cellular damage throughout the body, creating something of a vicious cycle where mitochondrial dysfunction contributes to cellular aging, which in turn further impairs mitochondrial function. This mitochondrial decline has been connected in research to a wide range of age-related changes, including reduced muscle strength and endurance, since muscle tissue is particularly dense with mitochondria given its high energy demands, cognitive changes, since neurons similarly depend heavily on efficient energy production to function properly, and broader metabolic changes including reduced insulin sensitivity, since mitochondrial function plays a significant role in how effectively cells respond to insulin and process blood sugar. Given how central this decline appears to be across so many aspects of aging, researchers have invested substantial effort into identifying interventions, including specific dietary components that appear capable of supporting mitochondrial number, function, and resilience against this age-related decline. Research that has produced some genuinely well-supported and practical findings. The first food is fatty fish, particularly varieties rich in omega-3 fatty acids like salmon, mackerel, sardines, and anchovies, and the research connecting these fatty acids to mitochondrial health centers on a few distinct but related mechanisms. Omega-3 fatty acids, particularly the ones called EPA and DHA, found concentrated in fatty fish, become incorporated into cell membranes throughout the body, including the specialized inner membrane of mitochondria themselves, and research has found that this incorporation appears to influence mitochondrial membrane fluidity and function in ways that support more efficient energy production. Multiple studies examining omega-3 supplementation or increased fatty fish consumption have found associations with improved markers of mitochondrial function, including in research specifically examining skeletal muscle tissue, where mitochondrial density and function are particularly consequential for physical capacity and strength, both of which decline notably with age-absent intervention. Beyond this direct membrane-related mechanism, omega-3 fatty acids have well-established anti-inflammatory properties, and research has increasingly connected chronic low-grade inflammation, sometimes called inflammaging when discussed specifically in the context of aging, to accelerated mitochondrial dysfunction, since inflammatory signaling molecules have been found in laboratory research to directly impair mitochondrial function through several pathways. This means fatty fish appears to support mitochondrial health through at least two distinct and complementary routes: direct incorporation into mitochondrial membranes, and reduction of the inflammatory burden that otherwise contributes to mitochondrial decline. A combination that helps explain why research examining fatty fish consumption specifically, rather than omega-3 intake through supplements alone, has often found particularly consistent associations with markers of healthy aging across multiple studied populations. The second food is walnuts, and while nuts more broadly have shown various health associations in research, walnuts specifically have attracted particular attention in mitochondrial research due to their unusually high concentration of a plant-based omega-3 fatty acid called alpha-linolenic acid, along with a range of polyphenol compounds not found in comparable concentrations in most other commonly consumed nuts. Research examining walnut consumption has found associations with improved markers of oxidative stress, directly relevant given what we discussed earlier about aging mitochondria producing increasing amounts of damaging reactive oxygen species, suggesting walnuts may help offset some of this age-related oxidative burden through their notably high antioxidant content. More specific laboratory research examining walnut extract and isolated walnut compounds has found effects on mitochondrial biogenesis, the process by which cells generate new mitochondria, a genuinely significant area of interest for aging research since supporting this process could theoretically help offset the age-related decline in mitochondrial number discussed earlier, rather than simply protecting existing mitochondria from further damage. While human clinical research specifically confirming this biogenesis, promoting effect from whole walnut consumption remains an area of ongoing investigation. The combination of walnuts' plant-based omega-3 content, substantial polyphenol concentration, and the laboratory research suggesting effects on mitochondrial generation processes has made walnuts one of the more consistently recommended foods within nutritional research specifically focused on cellular and mitochondrial aging. The third food is leafy green vegetables, including spinach, kale, and Swiss chard. And the mitochondrial relevance here connects to a nutrient that receives considerably less popular attention than vitamins and antioxidants, but that research has identified as playing a direct and essential role in mitochondrial energy production itself, namely magnesium. Magnesium serves as an essential cofactor for numerous enzymes directly involved in the mitochondrial process of generating ATP, meaning adequate magnesium isn't simply generally supportive of health in some vague sense, but is mechanistically required for mitochondria to efficiently complete their core energy-producing function. Research has found that magnesium deficiency, which population studies suggest is relatively common, particularly among older adults, is associated with measurably impaired mitochondrial function and reduced energy production efficiency. And separately, magnesium status has been found in research to naturally decline somewhat with age due to reduced absorption efficiency and other age-related changes in how the body processes and retains this mineral, making adequate intake through diet particularly relevant for older adults, specifically working to support mitochondrial health. Beyond magnesium, leafy greens are rich sources of nitrates, discussed extensively in other research contexts for their role in nitric oxide production, and research has found that adequate nitric oxide availability, beyond its well-known cardiovascular benefits, also appears relevant to mitochondrial function, with some studies finding that nitric oxide signaling influences mitochondrial biogenesis and efficiency, adding another mechanistic layer to why leafy greens show up so consistently across different areas of aging-focused nutritional research, supporting multiple interconnected biological systems through overlapping but distinct pathways. The fourth food is berries, particularly blueberries, which have received substantial specific research attention within mitochondrial aging research due to their high concentration of anthocyanins, the polyphenol compounds responsible for their deep color that we've discussed in other contexts as well. What makes blueberries particularly notable within mitochondrial-specific research is a body of laboratory and animal studies finding that blueberry compounds appear to activate certain cellular pathways involved in what researchers call mitochondrial biogenesis, and what's sometimes referred to as cellular stress resistance, essentially the cell's ability to withstand and adapt to various forms of stress, including oxidative stress, through mechanisms that appear to involve activation of specific cellular signaling pathways connected to mitochondrial health and resilience. Some of the more notable research in this specific area has examined effects on brain tissue, where studies in animal models have found that blueberry supplementation is associated with improved mitochondrial function within neurons, and correspondingly, improved measures of cognitive performance, findings that have generated substantial interest given how directly neuronal mitochondrial function connects to the cognitive changes associated with aging discussed earlier. While translating findings from animal research directly to expected human outcomes always requires appropriate caution, this body of laboratory and animal research, combined with the broader and more extensively established human research on berries and general markers of oxidative stress and inflammation discussed in other contexts, provides a genuinely coherent picture supporting blueberries and related berries as a meaningfully evidence-backed component of a mitochondria supportive diet. The fifth food is extra virgin olive oil, the primary fat source of the Mediterranean dietary pattern that has appeared repeatedly across so many different areas of aging and disease prevention research. And its relevance to mitochondrial health specifically connects to a combination of its healthy fat composition and its notably high content of specific polyphenol compounds not found in more processed olive oil products or in most other commonly used cooking oils. Research examining extra virgin olive oil specifically, as distinct from more refined olive oil products which lose much of this polyphenol content during processing, has found associations with improved markers of mitochondrial function and reduced oxidative stress, with some research specifically examining a compound called oleuropein, along with related polyphenols, found concentrated in high-quality extra virgin olive oil, finding laboratory evidence of these compounds supporting mitochondrial biogenesis and protecting existing mitochondria from oxidative damage. Population research examining Mediterranean dietary adherence, in which extra virgin olive oil serves as the primary fat source, has found consistent associations with markers of healthy aging and reduced markers of oxidative stress and inflammation throughout the body. And while this broader dietary pattern research reflects the combined effect of many foods rather than isolating olive oil's contribution alone, more targeted research specifically comparing extra virgin olive oil to other fat sources has found reasonably consistent evidence supporting its particular polyphenol content as a meaningfully distinct contributor to these broader mitochondrial and aging-related benefits, beyond what would be expected simply from its favorable fatty acid composition alone. Stepping back from these 5 specific foods, it's worth addressing how they work together within an overall dietary pattern, since research on mitochondrial health increasingly suggests that these different foods and their various protective compounds appear to support mitochondrial function through genuinely complementary rather than redundant mechanisms. Direct membrane incorporation from fatty fish, biogenesis support from walnuts and berries, essential mineral cofactors from leafy greens, and polyphenol protection from olive oil, together addressing multiple distinct points within the complex biological processes governing mitochondrial number, efficiency, and resilience against age-related decline. This combination-based picture aligns with a broader and increasingly well-established finding across aging nutrition research generally, that overall dietary patterns emphasizing diverse whole foods tend to show stronger and more consistent associations with healthy aging outcomes than any single food or isolated nutrient studied in isolation. A pattern researchers believe reflects the genuine biological complexity of processes like mitochondrial aging, which involve numerous interconnected pathways, unlikely to be fully addressed through any single dietary intervention alone. Beyond diet specifically, It's worth briefly noting that research has found regular physical activity, particularly exercise that meaningfully challenges the cardiovascular and muscular systems, to be one of the most robustly evidenced interventions for supporting mitochondrial biogenesis and function across the aging process, with numerous studies finding that regular exercise training measurably increases mitochondrial density and efficiency, even in older adults, suggesting that the dietary strategies discussed throughout this conversation work best not as standalone interventions, but as complementary support alongside consistent physical activity, together representing a genuinely evidence-backed, multi-pronged approach to supporting the cellular energy production systems that underlie so much of what we experience as healthy versus unhealthy aging. What makes this entire area of research particularly meaningful is how directly it connects dietary choices most people can realistically incorporate into ordinary life. Fatty fish a few times weekly, a regular handful of walnuts, generous servings of leafy greens, berries incorporated into meals or snacks, and extra virgin olive oil as a primary cooking and dressing fat, to biological processes occurring at the most fundamental cellular level throughout the entire body. Mitochondrial health may not be a topic most people think about during a typical grocery shopping trip, but the growing body of research connecting these specific, accessible foods to the cellular machinery responsible for essentially all of the energy that powers human life offers a genuinely compelling and practical framework for thinking about healthy aging. One grounded not in vague wellness trends, but in an increasingly detailed scientific understanding of what's actually happening inside our cells as the years accumulate. It's worth touching briefly on how researchers actually measure mitochondrial function in living tissue, since this context helps explain the confidence behind these dietary findings. Techniques used in this research range from muscle biopsies that allow direct microscopic examination of mitochondrial density and structure, to more indirect but still meaningful measures like exercise capacity testing, since how efficiently the body uses oxygen during physical exertion closely reflects underlying mitochondrial efficiency, to blood-based markers of oxidative stress and inflammation that, while less direct, correlate reasonably well with mitochondrial health status across large population studies. This combination of direct tissue-level research and larger-scale population and blood marker studies is part of why researchers have developed reasonable confidence in the dietary associations discussed throughout this conversation, since findings from more invasive, smaller-scale mechanistic studies have generally aligned with the patterns observed in larger population research examining these same foods and dietary patterns. It's also worth acknowledging that mitochondrial aging, like most of the biological processes discussed across this broader area of aging science, doesn't happen at a uniform rate across all individuals, and research has identified genetic factors, baseline activity levels, and existing metabolic health as all influencing how quickly or slowly mitochondrial decline tends to progress in any given person. This variability doesn't undermine the value of the dietary strategies discussed today, but it does reinforce why researchers in this field generally frame these foods as genuinely supportive interventions, capable of meaningfully influencing the trajectory of mitochondrial aging, rather than as tools capable of completely halting or reversing a process that reflects such deeply rooted and individually variable cellular biology. Taken together, what this research offers is a picture of healthy aging that feels both scientifically grounded and genuinely achievable, incorporating fatty fish, walnuts, leafy greens, berries, and extra virgin olive oil regularly into an already balanced diet, alongside consistent physical activity, represents a realistic and evidence-supported way to actively support the cellular energy systems that quietly determine so much of how vital and capable we feel as the decades pass. A reminder that some of the most consequential biology in the entire aging process is happening at a scale far too small to see, yet remains genuinely responsive to choices made at the dinner table.